My presentation will be jumping exactly into the matter of the topic. A few words about me. Indeed, I'm now Professor Emeritus, but I'm not retiring from science. I'm doing some projects. I'm very thankful to the board of directors of Helmholtz Zentrum Munich that I'm allowed to finish some projects which are funded externally, and I will be developing new projects in Singapore, and as well I'm phasing out as a co-founder to a small company, Metabolite Diagnostics. I was confronted with different types of cohorts in my work as a biochemist and the guy who was promoting the mostly analytics and as well data integration. So we are dealing in the analysis of cohorts with different type of cohorts. As a matter of fact, the cross-sectional cohorts were probably the largest so far, and they have some advantages, but the issue is if you are focusing on a specific phenotype, it might be underrepresented. Disease focus or case control cohorts are very facilitating development of understanding how the mechanisms of disease are present, the therapies, what are the biomarkers, but usually they have not too many individuals. Genetic effects or epigenetic effects cannot be properly analyzed. So I'm looking forward to that, what was already touched in the previous presentations, that we have to develop prospective large cohorts, and from the infancy to being adult and even senescent. These are at present quite rare, and only those would provide the longitudinal samples of individuals and a completely new window to the analysis of phenotypes. Most of the data presented today were derived from disease-focused or cross-sectional cohorts. Now, what I think is an essential collection of data which should be represented in the cohort. Well, I think the wide representation of phenotypes. I'm referring to the disease and not disease status, but as well other parameters which might be showing up as non-relevant to the disease. In the analysis to come, they might be very pivotal. A very critical effect, which I have seen many times, and I will show you an example for that, is how the cohorts are collected. The metabolomic cohorts or general cohorts are profiting from the biobanking, and this is no longer very much an issue, but we do see the effects of that still. Definitely, the thinking about the future, that multiple aliquots should be collected at the time of collection, that the throwing and the freezing, it might be an issue to the integrity of sample, whatever you do, whether it's proteomics or metabolomics. Mirrored safe storage is an issue everywhere in the world, and the blackout or earthquake can destroy such a biobanking, and this would be really a huge loss to science. Comprehensive additional phenotyping means rather analyzing the biometric data and differs to clinical phenotypes. It is as well the information about our exposition to environment and lifestyle. What is obvious, but this is not always present, this is how the biobanks are accessible and how are they managed. Is it easy to contact the people and deliver the data from the storage, or is it something which is closed? Now, the data protection is something which is getting a lot of attention around, and as well activation of patient cohorts or patient organizations, and as well consent of the patient is extremely important for the quality of the cohort, because it simply motivates people to contribute as well. Now, in my laboratory so far, we were happy to work with different approaches, and many talents of our students and postdocs and senior scientists were implemented here. We were happy to work with targeted metabolomics, and we used a lot of products from Biocrates from the very beginning of existence of this company. And we are able to measure metabolites here depicted by names, and the number of metabolites is given to the right. We are as well happy to run lipidizer, which is a collaboration between SciEx and Metabolome, targeted analysis as well, over 1,000 lipids quantified. So these are quantifying all methods. And for these methods, we have developed a lot of processes and descriptions how the different matrices can be processed. So that's a very versatile tools from cell culture to different tissues, rodent and human body liquids. And we're using as well Chrome systems for the sake of simplicity and price. They are very affordable in cell culture, for example, although this is being used for the diagnostics of newborns. Helmholtz has developed here a Markite assay for oxylipins, eicosanoids, which is very well working in the assays we have performed, especially in studying asthma, the lipid metabolism. Now we are as well using several generations of metabolome technologies, and altogether this illustrates a portfolio one laboratory can develop. But of course, this does not have to be. And why I'm showing this slide is that we were confronted in our collaborations with the question, which assay shall I use if I'm going to perform a specific session? This is not very easy to answer. It's simply the situation in which you can think about if you are just generating the hypothesis and you have no idea what's going on. For example, a simple knockout of a mouse, which is a silent, you don't have any clue. I would go for non-targeted metabolomics. Another story is if you are looking for a quantification of changes which are going over the time and would like to have a very clear information about the absolute concentration, then I would go for the targeted metabolomics. But this has to be balanced from case to case, and this works in the hands of many people. Just an example of a SOP issue, which I still do see. Imagine before we start large cohort analysis, we perform a small set of measurement. This is an example from targeted analytics of one sample collected in one type of sample collected in one city. And the previous principal component analysis showed some distribution, and we had very good knowledge about the concentration. It looked to us very well. Fitting to the reference values we know. Maybe this is outlier, we don't know. This is maybe an effect of phenotype. But the moment we plotted collection sites, we notice that there's something going on here. So it means this is the same city, the same phenotype, but these samples were collected in two different hospitals. So even if the SOP is communicated to the collection site, there might be still a problem. Now, going further into the application side, you may imagine that there is always a large question: how can we explain a phenotype which omics is producing the most of the information? I'm citing a paper here, not of my own. This is the paper by Cirulli. Greg Venter was as well a co-author of this manuscript. The idea was to find out which information is the most important for explanation of the BMI. In the central figure here, you have a BMI transformed to normality versus metabolome represented in different individuals. And this different four types of BMI, from healthy to quite unhealthy, is depicted here as well. And the general message here is that if you are looking at metabolites collected here with non-targeted metabolomics and you compare it with a phenotype, genetic component is less than 4%. And metabolomics explains more than 50% of the phenotype. So it was a quite big surprise to me that this amount can be reached. But this is how the metabolomics can provide as a proxy to the explanation where the metabolite comes from. Maybe even if we don't have the gene, we may have the pathway. Now, something from the very recent publication I was happy to contribute just came out in Nature in December 2020. This is another study in which the metabolite in the human blood was analyzed, and the question was if we can explain the variance in metabolites by diet, by clinical data like blood pressure, for example, microbiome, genetic background, circadian rhythm, and lifestyle. And this is a level of explanation. This is a number of metabolites which explain the effect. And as you can see, if you combine several models, you can get quite pretty nice explanation. But the message from this slide is that the genetics and circadian rhythm apparently in this population are not contributing as much as the diet and clinical data and microbiome. Of course, you know already that there is interaction between the genetics and clinical data, that our nutrition is impacting our microbiome, so they are not really independent. But definitely here, the same story is shown that the diet and microbiome and clinical aspects, they are explaining a lot of our metabolic variation in the blood. Now, I would like to convey you a message in which I am facing a problem very often if we are discussing the data integration between different omics data sets. This is very obvious to me that if we are talking about the genome sequencing, we are dealing with actually only 4 levels of freedom, 4 degrees, A, T, C, G. If you know them, you can calculate the indels, SNPs, copy number variations, and of course, not from that sequencing, but maybe indirectly, you can as well derive the epigenetic information. But the 4 levels of variance are given here. Now, if you are talking about the metabolomics or metabolite, you have a much more levels of freedom than the 4. You have presence, you have concentration, you have tissue, yes or no, you have time, you have pathway. So a lot of them, they are linear, like concentration. So the number of possible variations in the metabolome is much higher than in the genome. Therefore, I would give you this advice, or maybe my experience is that distinct omics approaches develop in different methods. Right? They are not really to be exchanged. They don't simply work. And especially for metabolomics, we need specific study design and specific stratified randomization. Let me give you an example for that. So first of all, contrary to the genome sequencing, you should not randomize during the measurement the longitudinal samples derived from one individual, because the equipment drift whatsoever around it may affect the batch, and the batch effect might be larger than the longitudinal changes. So these are not randomized. Some longitudinal samples are not randomized. The best is as well to have randomization for study center, control versus disease, and confounders and so on. So if you have one plate of controls and one plate of disease, then definitely you will find differences because of batch effects and not because of a biological case. The best is, of course, to randomize the hypothesis you are going to test. This is good if you have already an idea how to perform the measurement, which hypothesis is to be done, because if you are going to go retrospectively, of course, you cannot repeat the measurements over and over again. So multiple randomizations are the choice. I just give you an example from the real study. If you imagine a large consortium with several study centers running SOPs and perform a metabolomics, targeted metabolomics, several thousand samples, and just plot, color code the origin, and you see that these colors are not really overlapping. At this moment, you don't know whether it's a phenotype or, but it looks at the centers would be completely different. So maybe they have different phenotypes. Now, if you perform the phenotype coding, you see that there is something different shown here than here, and now suddenly you don't know where is the phenotype difference coming from. Is it from the center or is it from the biological background? You have to find it out. Now, let us have a case study here. So define hexose as a phenotype definition. The control does have some low level of hexose, and higher hexose, in this case glucose, will be a definition of manifest type 2 diabetes. Okay, so this is our definition, how to differentiate the control case. Now, we have on this slide two different ways of a randomization of samples. Now, on this one, we're using randomization for the reference and linear model. Okay, so this would be approach which could be used in the metabolic-oriented study. As you can see here, after the several steps, you have some representation of phenotype, and it looks like we are having a good starting point for the further analysis. If you randomize it as it would be for the genome sequencing, for example, it means you are randomizing everything with everything, barcode, plate, number of sample, study center, whatsoever, and then you are receiving this type of appearance. It looks as there would be very little phenotype difference. Okay, the coding is for the phenotype. How to prove it? We are going back to hexoses. So without barcode code, you retain your phenotype. But if you're using barcode approach, I mean randomization for everything, you are losing the phenotype. So the type of randomization could contribute a huge bias already before you start measuring. Now, what I think is very important in the cohort to perform nice cohort studies. So first of all, we have to provide a really comprehensive metabolite coverage. I mean, hundreds of metabolites have to be quantified. The analytics must be reproducible. The QCs have to demonstrate how robust is the analytics, that the metrics is working. It must be sustainable. So if you come back to the assay a few years ago, you have to have tools allowing you to see what are the differences, and if the samples are identical, must be accessible to everybody, and not only to selected few people in the world, and affordable, of course. And it should match the throughput. Throughput is growing everywhere in the world. The standardization and scalability, it's a new word which is maybe not standardization, but being scalable is something which has to be addressed once again and in a different way. So there are different ways to do that. First of all, the metabolites are being measured in two different aspects today. It could be grouped in targeted, non-targeted metabolomics. As I told you, we were doing already a lot of research with Biocrates and Metabolon, and this is an example of a study in which the same sample was measured with untargeted metabolomics and targeted metabolomics. There is an overlap. So you can see here named metabolites. Out of those who were measured, it's about 800 in this particular assay. This was 141. But what I observe for development in the targeted metabolomics, that the number of metabolites we can measure is growing constantly. So there's a new product from Biocrates, MXP Quant 500, which is allowing already quantification of over 600 metabolites, okay? Lipidizer is allowing quantification of 1,000 lipids already. So this is a kind of decision you can make already in a study design. Where to go is as well which method to choose. Standardization of assays can be done in different ways. A classical ring trial is derived from the laboratory medicine, where you have one assay and you go around the world through different laboratories and observe only the absolute value which is being analyzed. This is very important to see if there is a bias in different laboratories, and if they then measure the different cohorts, how does it affect the data interpretation? There are recently as well ring trial which are devoted to chemical classes, and this is usually for classes which were not covered yet in in a large extent. There are as well some attempts to describe all metabolites in one sample, I mean several thousands of metabolites, just to have a view what is already present. The sample is distributed then among many laboratories running different technologies. All this contributes to the standardization of metabolomics. Some examples: we were happy to contribute in several ring trials in metabolomics, which brought us to compare how the same assays performing different laboratories worldwide. This absolute IDQ P180, very successful for large cohort studies. There's a new ongoing trial which I am coordinating right now. All measurements have been finished. We are starting the evaluation. There is another ongoing trial on ceramides and bile acids coordinated by Markus Benck from National University of Singapore. A lipidizer ring trial coordinated by Martin Gera. And they all contribute to the standardization and scalability of metabolic assays. So what is the output? The output is then that you have the concentration of a given metabolite with the given variance, which is caused by the single laboratory. You can as well have this type of presentation in which the known concentration in the sample, which is worldwide available, like this NIST 1950, is compared to the performance for the given metabolites, and this is being correlated between the information known from the NIST sample and the information measured in the assay. So this is as well a kind of standardization. Now, why I'm telling you that? We are at present in situation in which people have cohorts, and these cohorts, they are simply undergoing several assays, analyses, okay, this in the red. Let's pick one assay, could be targeted metabolomics, and the cohort is maybe 4,000 or 5,000 samples big. This is very reasonable to measure it within a reasonable amount of time, several months, maybe 1 year, depending on the equipment present in the laboratory. This has been demonstrated several times as well by speakers today in the meeting, but we are moving to another situation. And now my cohort is 40,000, 200,000, 500,000 samples to be measured, not in 10 years. It has to be measured within a reasonable time, maybe 1 year, something like that. So this is the issue of scalability. Only few products on the market are able to support this, like MX 500. This assay is able to support it. So this is why we are doing this rig trial, to have a standardization worldwide, to provide the possibility that many laboratories are measuring the same cohort, and this large cohort is being processed very fast at a high quality. The same may happen that there another cohort, which is maybe 100,000, would like to be compared with the other one. So other laboratories in another country, they could use the same technology, and then the ultimate replication using the same molecules, the same metabolite measured, absolutely could be done. Of course, it is possible to send it to one laboratory and spend millions of dollars on that. This has been shown to be very successful, but we are now thinking about different dimensions. This is something which is worldwide asking for new solutions. And I think this scalability, the assays which are being used parallel in different countries, different laboratories have to be considered. Now, what I think are future strategies showing up along the metabolomics and cohorts. I think the longitudinal GWAS or metabolite-wide association studies will be a very active component, especially if we are thinking about the analysis of individuals over several years with several samples per year. This is something which was not done. Environment interaction, especially microbiome, is something which is exploding right now. Standardization tools in assays like ring trials, like using the same standardization or reference samples. This is which is developing very well. Parallel processing in multiple labs using the same assays is something which I'm suggesting many people will have the same idea to use for processing of very large cohorts. What I'm missing is something like the definition of a meta description of data, which has to be reported before the manuscript is even published. I see great papers with a huge depth of data in it, and these manuscripts are not really comparable because there's always something missing. High impact journals now have the rule that the published data are open access to everybody. Well, this is not the case in every other journal, and I think genomics is far more developed in this direction than the metabolomics. Data protection implementation is something which has to be considered because using the genome is the best way to identify the individual, but a metabolome will tell you as well what happened to the individual, and then you may derive some social critical information about that. So this is something to be considered. A few words about the longitudinal challenges for the metabolomics. I think the investment in future biobanks started. We have seen today some information derived from cohorts which have a limited set of longitudinal samples, sometimes only a few years apart, so maybe 2 time parts. This is Epi, Cora, and Carla, for example. Some have different background, like migrants moving from one country or one continent to another one. Great information derived, but this is completely different to that what the native populations would like to see. Some have been developed for, like phase in Singapore, and they have some samples, but the number of them is quite limited. Today we had information about the new cohorts which are developing, and this is the future where this longitudinal analysis could be done. So the Singapore, the national cohort, the Tohoku University, Korea Biobank, these are new initiatives. What I think is worth exploring, it has been mentioned today several times, is the inborn errors of metabolism. I think the nature had made these knockouts, these mutants in humans, and most of them are lethal. Over 8,000 monogenetic diseases with different penetrants are known so far. We are missing many because we don't know that they are there. And many syndromes like neural retardation development syndrome, after introduction of exome-wide sequencing, have been differentiated to very distinct specific diseases like poor alpha mutation in transcriptional factor. Now, the question is, if we know the longitudinal axis, how many of these errors of metabolism temporarily affected, only present in several, in the time window, which is in the youth or in the elderly? So this is a completely unsolved question. Now, I think the identification of genetic component in the longitudinal response is as well worth still discovering. We know that the raw DNA sequence is not explaining frequent diseases like diabetes, like neural disorders, like cancer. If we think about the epigenome metabolome analysis, epigenome proteome analysis, I think this longitudinal component will bring completely new discoveries. A few words about the microbiome. As you may know, we have more than 1,000 species in our body. They represent genomic richness, which is 100-fold more than that of human, and they have been already demonstrated of key importance to our health. However, the recent information is as well contributing to the knowledge that the specific signatures are not only seen in stools or feces, they are as well seen in blood plasma, and in animal models you can see them as well in tissue. So indications which have been already described and discovered using microbiome metabolome analysis cover diabetes, diabetes type 1, diabetes type 2, non-alcoholic fatty liver disease, neurological disorders, cancer, atherosclerosis, immunity, pharmacokinetics, and as well, I would like to mention my last cell report paper done in collaboration that energy balance as well is being heavily modulated by the cofactors provided by microbiome. So we are talking about the whole communication system affecting brain, our body and liver through specific metabolites. And this is what we need in the future, that we need assays which are covering bile acids, tryptophan metabolism, short chain fatty acids, indole steroids, brain chain amino acids, lipids, ceramides in one assay. Biocrates has one assay covering that. You can do a lot of work with them. Now I'm at the very end, and I would like to say some things to my collaborators in the last few years. First of all, I would like to express my gratitude to my research group, where we have created so many metabolic tools, metabolomic tools. And as well as I would like to thank Gabi Müller for her patience with my personality. Would like to thank Karsten Sule and Ania Halama. Hope she's online. Dziękuję bardzo, Aniu, for all collaboration and inspiration. The metabolomic at Helmholtz wouldn't be at the place it is now without the collaboration with Annette Peters. Thank you very much, Annette. With Christian Giger, Rui van Zandler, Sapna Sharma, and many other colleagues. And we were using cohort as a startup and then diverse to many others. We'd like to thank Gabi Kastenmüller and Werner for the continuous support in data analysis and bioinformatics. Many thanks to Ken and Kirsten for the circadian rhythm studies. I was not able to present them here today, it would be too much, but this was a nice piece of work. Now I would like to thank my collaborators from Japan, Professor Hironobu Sasano, for the collaboration in steroid signaling pathways, and Professor Saizo Koshiba and Daisuke Saigusa for the collaborations and discussions and open access to discussions and data in their laboratory. I would like to thank as well great team from Biocrates to the founder, Bert Rocha, previous CEO, Wolf Fischer-Knuppert, and present CEO Nick Schauer. We had great discussions about strategies. And thank you very much to the team who organized the meeting today. I'm very happy that in the past years we were working on different aspects of science, presentations, kit development as well. I'm very thankful to John Riles, Annie Evans, and Edward Caroli for discussions. It was very fruitful. Now at NUS, National University of Singapore, I would like to thank to Marcus and Anne, Mauri and Federico Tota for the collaborations we are doing right now, as well for the work I have done with Joachim and Drosophila and Microbiome and Metabolite plasma, explaining the best phenotypes using metabolomics. This is work done with Weizmann Institute of Science, the office right now in the United States, and MTI. Todaraba. Then the biggest project I ever been contributing, almost 10 years of IMI Direct. I would like to thank the guys from the clinical science, explaining to us the phenotypes in one person, Hartmut Rutten, Paul Franks, talking about the clinics and epidemiology, Oluf Pedersen about microbiome contribution, Anna Venuela, Mark McCarthy for the genomics, Johann Schwenk for proteomics. From Bernd Jablonka, I've learned a lot about project monitoring, and Imre Pavlo for the interactions industrial project monitoring. Gary Frost for the nutritional science. So these not complete list, but I think the last few years were very successful, and a lot of good manuscripts came out, and a lot of training and connections have been created. At the very end, I would like to thank to my tutors, to my friendly advisors, to people to whom I went when I was in trouble, and the doors were open to me, and they have advised me what to do and what not to do. And very, very great thanks to you guys. Wish you all the best. At the very end, if you're interested in some stuff which has been discussed today and would like to read something about it, there is a book just published by Elsevier. The book is on metabolomics for biomedical research. I have edited it and as well co-authored that. And Augustin Scalber is as well a co-author. Merci beaucoup. If you Google metabolomics for metabolomics research, I'm sure you will find this resource which could support your research. Thank you very much for your attention.